Hi, so we're still looking at problem solving. This is the experimental design question from the SQA 2013 paper, question 4. Okay, here's our context. In an investigation into the effect of lead ion concentration on respiration in yeast, 2 flasks were set up as below. So we have just a description in a wee table. Flasks were placed in a water bath for 20 degrees C for 10 minutes. And after this time, 2.5 centimeters cubed of yeast suspension was added. Oxygen sensors were fitted. And they've got a little diagram for this one. The flasks were left for a further 10 minutes and then oxygen concentration was measured in each flask every 20 seconds for 2 minutes. Here we go. Right, so we've got our time going up to 120 because that's our 2 minutes because it's in seconds. We've got Flask A with 0.2% lead nitrate and Flask B with 1% lead nitrate. And what we're looking at is the oxygen concentration in milligrams per litre, mgs per litre. Okay. Identify one variable not already mentioned which would have to be kept constant so that valid conclusions could be drawn. So a common question, they're looking for a variable not already mentioned. So let's have a look at what has been mentioned, and we're not going to be able to talk about them. So we have volumes of glucose solution, so we can't say anything about that. We've got volume and concentration of lead, so we can't talk about that. We've got temperature, we've got the minutes it was left for. We've got the volume of the yeast suspension, and then we've got how the measurements were taken and how long they were left for. Okay, so we need to work around this. So you've got to look for something which is in here but has not got more information about it. So, well, we've got volume of glucose solution, but we don't have concentration of glucose solution, so that's a potential. I think this is the one that most people will go with. The yeast suspension, we've got volume, but we don't have anything about the type of yeast, species of yeast, the age of the yeast, concentration of this. So this is probably your best one to go with. Okay, explain why the flasks were left for 10 minutes before the yeast suspension was added. Well, that one, if you look at the context, it was left for 10 minutes before the yeast was added while they were in the water bath. So it's to let it get up to temperature to make it ready for that. Explain why the flasks were left for a further 10 minutes after the yeast suspensions were added before the measurement of oxygen concentrations were taken. So this time we should be up at temperature, but you're now looking to have the effect happen on a cellular system. So you need some time for that to have an impact. So you're looking for time for the lead concentrations to affect the cells. Okay, so we have a graph question. We're asked to draw a line graph to show the oxygen concentration in Flask A. It is in bold, so therefore it's important that you just do that. Using an appropriate scale to fill most of the grid. This is standard now, you will see this every time, and that means that you know it's going to be part of the mark scheme, that if you don't do it properly you're going to lose it. Okay, so we're only looking at the time and the Flask A concentration in mgs per litre. Okay, Flask B we don't need to worry about at all. Okay, so you first of all you have to be sure you've got your axes around the right way. I quite like the dry mix, which is dependent responding is on the Y, measured independent is on the X. Or there is also the nappy rule. The nappy rule says what you changed goes across the bottom. Okay, so this would be time. And up the side we've got oxygen. Just lift the whole title from the table. Doesn't matter if it's huge, lift the whole thing. Not so good going sideways on this one. Okay, we're looking for a 0 on both axes because this one's got a 0, this one's got a 0, so put the zeros in. Okay, on the time we need to get up to 120. So what would that be? 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 110, 120. They've given you it perfectly. So you fill in every single check mark. Don't miss any of them off and make sure that it's a little bit neater than I'm managing to write just now. Okay, up the side we need to get up high enough to get to 10.2. So if this was 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, they've given you a really nice scale. So that's going to be just going up like that. So every division here is 0.2. Okay, you're not going to write that in because then you'd have to write every single one of them in as soon as you put the check mark on. So your first point is here. Your 10, oh sorry, that's not true. Let me just wipe this bit off. Your 0 is 10.2 up here, very neatly on the line, better than mine hopefully. 8.4 and so on. Okay, it requires you to be within, they expect you to be able to deal with a half box, so that 6.1 has to be between the two lines. If you don't do that, that's not a correct point. 3.8, we're going 6 is 3.8, 3.8, so there. Sorry, mine are still not perfect. 80, so again on the 1.7 you need to be halfway between the two lines, and the 100 is 0.2 and this is 0, and we're looking for neatly joined lines, which I cannot do on this, so you would do it neatly. Okay. Okay, using information from the table, state the effect of increasing lead ion concentration on the aerobic respiration of yeast. This is actually, I think, pretty straightforward. We are going from 0.2 here to 1, so this is increasing lead concentration, and the oxygen concentration is here, so you'll see that in 0.2% our oxygen concentration drops to 0, but in 1% it only drops to 3.2. So increasing lead ion concentration decreases aerobic respiration of yeast. That's it. Bubbles of gas appear in both flasks throughout the investigation. Name this gas. Well, you really should know this one by now. This is what we get from respiration. Absolutely acceptable to write CO2. It's not okay to write that, because that's not CO2, and it's not okay to write that, that's not CO2 either. Okay, explain why this gas continued to be produced in Flask A at 120 seconds. So Flask A, they ended up with zero oxygen left, but still was producing carbon dioxide. Well, that's all we know, that this is kind of preferred option because you're going to get loads of energy out of this, but as soon as you run out of this, cells will switch to anaerobic respiration, so fermentation pathways, and what you'll now have is the glucose will be just changed to carbon dioxide and ethanol in the case of yeast. So why have we got this? Because we now have fermentation taking place. Okay. Here's your mark scheme. Plenty of options, so make sure that you've checked that yours makes sense according to the mark scheme. This, again, you're going to have to be pretty brutal with your marking of graphs. They have to be done well. And that's another one done.